guys, welcome to the first part of studying pharmaceutical microbiology. And for today, we will begin with the overview of microbiology, what it is, how it impacts our lives, why is it essential in the pharmaceutical field. We will cover the definition of microbiology, explore how microorganisms affect us, understand the naming and classification of microbes, and take a brief look into the history of microbiology along with the key figures who shaped the field. Also, we will touch on the recent advances and discuss the importance of microbiology in modern science and healthcare. So, let's start. Microbiology is the study of microbes or microorganisms. These are tiny living organisms usually invisible to the naked eye. They include bacteria, fungi such as yeast and molds, protozoa, microscopic algae, and viruses. While viruses are not cellular and often exist at the borderline between living and non living, they are still studied alongside other microbes. A common misconception is that microbes are only associated with disease. But in reality, most microbes are not harmful, and they play essential roles in maintaining life and supporting human progress. In the environment, microbes help balance ecosystems by forming the base of aquatic food chains, decomposing waste, recycling vital elements, and fixing nitrogen in the soil. Photosynthetic microbes such as algae and cyanobacteria also contribute significantly by producing oxygen and organic matter. In human health, certain microbes aid digestion and synthesize essential vitamins like B complex vitamins and vitamin K. Commercially, microbes are used to produce vitamins, enzymes, alcohols, organic acids, and important drugs. The food industry also relies on microbes to make vinegar, cheese, bread, and alcoholic beverages. In biotechnology, microbial enzymes are used in making insulin, cleaning products, textiles, and more. Although only a small fraction of microbes are pathogenic or disease causing, understanding them is vital in healthcare to protect vulnerable individuals. Now that we know about microbiology and the ways that microbe affects us, let's now discuss how microorganisms are named. Taxonomy is the science of classifying, describing, identifying, and naming living organisms. The formal system of taxonomy was first developed by Carlos Linné, who laid the foundation for organizing biological diversity. Taxonomy involves three main components: classification, which is the orderly arrangement of organisms into hierarchical groups based on shared characteristics; nomenclature, which is the system of assigning standardized names; and identification, which involves determining an organism's traits for proper placement within taxonomic schemes. A key feature of modern taxonomy is the use of binomial or scientific nomenclature, which assigns each organism a two-part Latin name. We have the genus, which is the first letter of the name is always capitalized, while the species name is written in lowercase. And both are either italicized or underlined. For example, here we have Staphylococcus aureus. As you can see, the genus name Staphylococcus has the letter S capitalized, and the species name Aureus is all written in lowercase. This universal system ensures that scientists around the world can accurately communicate about organisms regardless of the language or region. Microorganisms, like all life forms, have evolved over billions of years through gradual changes in structure and function. This is a process known as evolution. The study of the phylogeny of organisms helps us understand their natural relatedness and evolutionary history. Phylogeny reveals how organisms are genetically connected and how they are diverged from common ancestors over time. Based on these evolutionary relationships, all living organisms are classified into three domains of life: bacteria, archaea, and eukarya. The bacteria domain includes true bacteria, which are unicellular prokaryotes with peptidoglycan in their cell walls. The archaea are also prokaryotic but differ significantly from bacteria. They are known for surviving in extreme environments such as high salt concentrations, acidic conditions, and intense heat. Finally, the eukarya domain also includes all organisms with a nucleus and a membrane-bound organelles, encompassing fungi, protozoa, algae, plants, and animals. This classification system reflects the vast diversity and evolutionary complexity of life on Earth. Now that we know all these things, let's proceed to the history of microbiology and the important people who contributed to this field. The field of microbiology began with the key observations in the 17th century. In 1665, English scientist Robert Hooke examined a thin slice of cork under a crude microscope and described the tiny compartments he saw as cells. This marked the beginning of the cell theory, which states that all living things are made of cells. Although Hooke's microscope could show large cells, it lacked the resolution to reveal microorganisms. That breakthrough came with Anton van Leeuwenhoek, a Dutch fabric merchant and an amateur scientist who is known as the father of microscopy. Between 1673 and 1723, he became the first person to observe live microorganisms using powerful, simple microscopes he built himself. He called these tiny creatures animalcules and recorded observations of bacteria and protozoa from rainwater, feces, and material scraped from teeth. His detailed drawings were the first accurate representations of microbial life. These discoveries sparked a major scientific debate about the origin of life, specifically the theory of spontaneous generation. It is the belief that life could arise from a non living matter. It was commonly thought that maggots arose from decaying meat or mice from soil. In 1668, Francesco Redi disproved this idea for larger organisms by showing that maggots only appeared in meat exposed to flies. With his experiment, sealed or gauze covered milk produced no maggots, proving that flies were the source. Still, many scientists believed that microorganisms could arise spontaneously. In 1745, John Needham conducted an experiment in which he boiled chicken and corn broths, then sealed the flasks. When microbial growth appeared, he claimed that microbes had developed spontaneously from the fluids, supporting the long standing belief. However, in 1765, Italian scientist Lazzaro Spallanzani challenged Needham's conclusion. He argued that microorganisms from the air had likely entered the broth after boiling, causing the contamination. To test this, Spallanzani boiled nutrient solutions after sealing the flasks, and no microbial growth occurred. His work provided stronger evidence against spontaneous generation, though critics argued that sealing flasks excluded oxygen, which was thought to be essential for life. In 1861, however, Louis Pasteur conducted a series of elegant experiments that finally disproved spontaneous generation of microorganisms. Using a specially designed swan neck flasks, he showed that broths remained sterile despite being exposed to air, so long as airborne microbes were prevented from entering. This work not only proved that microbes do not arise spontaneously, but also demonstrated that microorganisms are carried in the air. Beyond this, Pasteur made critical contributions to science and medicine. He developed the process of fermentation, showing that specific microbes cause the conversion of sugars into alcohol, and later created pasteurization, a method to kill harmful microbes in liquids such as milk. His findings laid the foundation of the germ theory of disease, proving that microbes are the cause of many illnesses and transforming both microbiology and healthcare. Was the period of extraordinary progress in the field of microbiology, marked by major discoveries in disease causation, immunity, and microbial techniques. It was led by pioneering scientists like Louis Pasteur and Robert Koch, whose work laid the foundation of modern microbiology and medicine. Louis Pasteur is also considered the father of modern microbiology. Again, he disproved the theory of spontaneous generation through his famous one neck flask experiment, supporting the concept of biogenesis, the idea that life arises only from pre existing life. Aside from this, Pasteur investigated disease in silkworms, identifying both fungal and protozoan causes. Robert Koch further proved the germ theory by demonstrating that Bacillus anthracis causes anthrax. He established the Koch's postulates, a set of criteria used to link specific microbes into specific diseases, solidifying the role of microorganisms in infectious diseases. In the realm of surgical practice, Joseph Lister applied the germ theory by using phenol or carbolic acid to disinfect wounds, dramatically reducing the post surgical infections. His work was inspired by Ignaz Semmelweis, who in the 1840s showed that hand washing reduced childbirth fever, an idea ahead of its time but crucial for aseptic techniques. Aside from that, Edward Jenner developed the first vaccine against smallpox, marking the birth of immunology. Paul Ehrlich contributed to chemotherapy by proposing the concept of magic bullet, a chemical that could selectively kill pathogens without harming the host. His work laid the groundwork for antimicrobial treatments. Also, Alexander Fleming discovered the first antibiotic, penicillin, a breakthrough that revolutionized the treatment of bacterial infections and ushered the antibiotic era. This era not only revolutionized our understanding of microbes, but also led to life saving advancements in healthcare, settling the stage for modern medicine and public health. The discipline now includes specialized branches such as bacteriology, mycology, parasitology, immunology, and virology, each focusing on specific groups of microorganisms. These advances have enabled critical developments in the environmental, agricultural, medical, and biotechnological sectors. For instance, sewage treatment processes now utilize beneficial microbes to purify water, while bioremediation employs microorganisms to clean up pollutants like oil spills, toxic waste, and chemical contaminants. In agriculture, microbes are used as biological pest control agents to treat crop damaging insects without harmful chemicals. In biotechnology, microorganisms are harnessed to produce food products, drugs, and vaccines, while genetic engineering allows scientists to manipulate microbial DNA to develop new therapies and bioengineered products. Microbiology also plays a vital role in the pharmaceutical industry through the development of antibiotics, vaccines, and diagnostics. Current applications span in food microbiology, agricultural and medical microbiology, and even the chemical and fuel production, highlighting the diverse and essential contributions of microbiology to modern science and industry. We have seen how curiosity, experimentation, and innovation unravel the mysteries of microscopic life. We explored the debate surrounding spontaneous generation, the establishment of germ theory of disease, and the critical role of microorganisms play in health, industry, and environment. As we move forward in the study of microbiology, we build upon this rich history and knowledge to better understand the invisible organisms that shape our world in profound ways, from causing diseases to solving global challenges. And this ends our first session in studying pharmaceutical microbiology, the overview of microbiology. And if you have questions, suggestions, corrections, or additional information, feel free to put them in the comments so that we can discuss it. Again, this is We Health Pharmacy. Join me as we study pharmaceutical microbiology. See you next time.